Good morning, students. Now I am going to discuss the topic CAM pathway. CAM means granulation acid metabolism. Granulation acid metabolism pathway is a special type of hormone fixation pathway. It is observed in plants that grow in dry, semi-arid or xerophytic condition. In order to survive in dry condition, plants adapt to the CAM pathway. Plants avoid photorespiration by using the CAM pathway. CAM pathway is found in Graciliaceae, Cactaceae, Agaveaceae and Orchidaceae family of plants. Plants like cacti, orchid and pineapple are found to exhibit CAM pathway. The stomata of these plants are observed to be scotoactive. Generally, plants lose their water content during the daytime through transpiration. In order to prevent water loss during the daytime, stomata remain closed and are open at night. In darkness, stored carbohydrates are converted into phosphoenol pyruvic acid by the process of glycolysis. The stomata in CAM plants are open in dark and they allow free diffusion of carbon dioxide from the atmosphere into the leaf. Now the phosphoenol pyruvic acid is carboxylated by the enzyme phosphoenol pyruvic acid carboxylase and is converted into oxaloacetic acid. The oxaloacetic acid is then reduced to malic acid in the presence of the enzyme malic dehydrogenase. The reaction requires NADPH2 which is produced in glycolysis. The malic acid produced in dark is stored in the vacuole. The malic acid increases the acidity of the tissues. During daytime, when the stomata are closed, the malic acid is decarboxylated to produce pyruvic acid and evolve carbon dioxide in the presence of the malic enzyme. When the malic acid is removed, the acidity decreases in the cells. This is called deacidification. One molecule of NADP plus is reduced in this reaction. The pyruvic acid may be oxidized to carbon dioxide by the pathway of Krebs cycle or may be reconverted to phosphoenol pyruvic acid and synthesize sugar by C3 cycle. The carbon dioxide released by deacidification of malic acid is accepted by ribulose diphosphate and is fixed to carbohydrate by Calvin-R C3 cycle. Significance of CAM pathway. In order to reduce water loss in CAM plants, stomata remain closed during the day and open at night. Using this adaptation, CAM plants survive in extremely dry conditions. CAM plants carry out carbon dioxide fixation in the dark. Hence, they are able to survive in light for a longer time without the uptake of carbon dioxide. When stomata remain closed, succulent plants obtain carbon dioxide from malic acid. CAM plants uptake carbon dioxide during the night. This limits photosynthesis. In addition, stored carbohydrate and organic acid also limit the photosynthesis process. Hence, the growth of CAM plants is generally slow. CAM plants are drought resistant. They possess xerophytic adaptation. Process of CAM pathway. CAM plants adapt themselves to survive under extreme drought or dry conditions. CAM pathway involves two important steps. First one acidification and second one deacidification. Acidification occurs during the night. In darkness, the glycolysis process converted stored carbohydrates into phosphoenol pyruvate. As the stomata of CAM plants remain open during the night, atmospheric carbon dioxide diffuses into the leaves. CAM plants fix the carbon dioxide using phosphoenol pyruvate. Phosphoenol pyruvate gets carboxylated and oxaloacetic acid is produced. This reaction is catalyzed by phosphoenol pyruvate carboxylase. The oxaloacetic acid is then converted into malic acid in the presence of malic dehydrogenase enzyme. NADPH produced during glycolysis is used for this reaction. Malic acid gets accumulated in the vacuole and causes an increases in the acidity of the cells. Deacidification. The deacidification process occurs during the day. In order to prevent water loss, stomata remain closed during the daytime. At this time, malic acid undergoes decarboxylation to produce pyruvate and carbon dioxide. This reaction is catalyzed by malic enzyme. As malic enzyme is decarboxylated to pyruvate, there is a decrease in the acidity of the cell. Therefore, this process is termed deacidification. Carbon dioxide produced during the deacidification of malic acid enters the C3 cycle and aid the production of carbohydrates. Pyruvate is either used for the regeneration of phosphoenol pyruvate or converted into carbon dioxide through Krebs cycle. CAM pathway is the most useful pathway in succulent plants. In order to reduce water loss, the stomata of these plants remain closed during the day. Therefore, atmospheric carbon dioxide does not enter the leaves during the daytime. However, these plants carry out photosynthesis during the day with the help of carbon dioxide produced by the decarboxylation of malic acid. Atmospheric carbon dioxide is fixed in the form of malic acid during the night. During the day, when atmospheric carbon dioxide is not available, stored malic acid is broken down to release carbon dioxide, which aids photosynthesis. Thus, CAM pathway conserves energy and aids the survival of succulent plants.